Yes.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh, and a very good evening to all of you. Thank you, Mr. Chairperson. My name is Muhammad Nashri Kimrani. I'm from University of Technology Malaysia. So currently I am a postdoctoral fellow in UTM. So before I begin my presentation, I would like to share small facts. I did my PhD 3 years ago. So Prof. Rosli was my supervisor. Another one important person sitting in front of me is Prof. Hisham. Hisham was my chairman for my Viva PhD. So suddenly I feel so nervous again. It's like I've been Viva again. So this is actually the part of my PhD studies back then. The integration of protein surface engineering with convolutional approach for chemoselective cross-linking of cyclodextrin glucanotransferase from social adhesives G1 aggregates immobilization. So a little background about the microbes. The source of the enzymes that we use is actually coming from the microbes called social adhesives. It's previously known as Bacillus lehensis. G1 is specific strain. This strain is locally isolated in Nanas, Johor. So this microbe is acarpilids. It actually grows at higher pH. The optimum pH is 10, and it is a good starch degrader. So this is actually how it actually degrades the starch. It actually produces the extracellular enzymes called cyclodextrin glucanotransferase, CGTase G1, which actually hydrolyzes the starch into the cyclic oligosaccharide known as cyclodextrin. So eventually, this cyclodextrin will enter the microbe and hydrolyzed by a second enzyme called maltogenic amylase to produce the prebiotic malto-oligosaccharide. So Prof. Hisham said about the prebiotic, so we actually produce the prebiotic as well. Okay, so this is the enzyme of interest, cyclodextrin glucanotransferase. It actually comprises of 5 domains. It actually catalyzes the starch to produce cyclodextrin. The cyclodextrin is very important as it is can actually form a supra-complex molecule with organic, inorganic compounds. So this particular sugar, cyclic sugar, is actually used as in perfumes, food, chemistry, pharmacies, medicine as well. Okay. So in order to improve the properties of this enzyme, we choose to immobilize it using the carrier-free cross-link enzyme aggregase immobilization. Is a carrier-free. So if you remember, this morning my supervisor, Prof. Rosli, talked about the carrier-free. He said that why are we using this immobilization? Because we are lazy. So it's actually talking about me. I'm the lazy one. So about this immobilization, the free enzyme aggregates the enzyme using the solvents. We use organic solvents or salts. After that, we just put the cross-linker to cross-link enzymes, and then we got the insoluble enzyme. So this insoluble enzyme we can use again and again and again. Okay? So, but the problem with this enzyme, this immobilization approach, is actually when we put the cross-linker is actually random cross-linked enzymes, and it actually produces some problems such as mass transfer diffusion, where when the cross-link actually crossing at the active site of the enzyme will actually close the active site of the enzyme, so the substrate cannot go in. So we want to try to improve this limitation by using the site-directed mutagenesis to actually reorientate and enhance the intermolecular interaction of this enzyme. So there is 3 method or factors that actually influence this approach, where we need to know the chemistry of the cross-linkers, the type of cross-linking, and to rearrange the enzyme. So we conducted our studies using the combination of the computational method with the rational design. We choose the site-directed mutagenesis. So this is the aim of our study. We want to do two things. First, to improve, to strengthen the intermolecular interaction, and second, to reorient the immobilized enzyme. Okay, so when we actually reorient, we want the active site face outside when it actually cross-linked, so that it actually discard the limitation of the substrate diffusion limitation. So we identify the region to modify the site reduction glucanotransferase using the conventional. We do the docking analysis first using the common cross-linker, glutaraldehyde. So we found that the domain D, which is actually located at the back of the active site, to be the most suitable regions to be mutated. So we constructed a total of 4 mutants. So this domain D we mutate to serine and lysine. Why serine? Because based on the docking analysis, we found that the glutaraldehyde binds to the serine amino acid. Why lysine? Because from the literature, we know that the chemistry of the aldehyde actually binds to the amino group of the lysine. So we try and we construct these 4 mutants to compare. So we do the homogeneity modelings to see the structural stability. So this is the homogeneity modelings of the mutants. And we see we validate the structure using ARAT and verify 3D. We found that all the mutants actually more or less the same with the wild type. And then we proceed with the cloning and transformation. All the total of 4 mutants we identified using PCR, using specific primers. We actually validate our clone using colony PCR and also sequencing. And then we later transform in E. coli as the special host. So first, before we immobilize, we see and check the free enzyme and free mutated kinetic. So the kinetic actually shows that this mutation has no effect on their performance. So we proceed to immobilize, and we do docking back to see whether the interaction of the cross-linker actually improve or not. And the summary is actually from the wild type, the glutaraldehyde that we first initially bind to the serine. When we mutate to the lysine, it actually bind to the lysine. When we mutate to the serine, it actually bind to the serine as well. But compared with these two, the serine actually has a higher binding affinity compared to the lysine. Okay? So we developed the immobilization. We tested with several precipitate agents, concentration of glutaraldehyde, type of cross-linking. We managed to recover 57.5% activity recovery using wild type first, and then we compare with our mutant, and we do increasing glutaraldehyde cross-linking concentration, and the figure is actually summarized. So when we increase the GA concentration, the glutaraldehyde concentration, we notice that when we use the wild type, it becomes compact because too much cross-linker. But in a mutation, it actually become bigger because the activity recovery is higher. So we know that something happened. It actually successfully enhanced the interaction and also able to reorient the immobilization. So we tested with different pH, mode of cross-linking. We come out with the mechanisms of the cross-linking. We conclude that actually the interaction happened at the carbon-oxygen, double carbon-oxygen between serine and the cross-linker. And then we proceed with characterization using PSAM, particle size, we notice that it's actually improved the thermal stability and pH stability as well. And then, interestingly, the kinetic performance is much higher than the wild type, where you can see the kinetic efficiency of the serine is 87 compared to the wild type is 32. And it actually also show the higher mechanical stability, the production of the cyclodextrin, and actually it can be reused up to 5 cycles, and it shows higher than wild type. Okay, so the conclusion from our study, we show that actually it's very important to determine the preferential amino acids between cross-nuclear and enzyme in organization. So we managed to show that for the glutaraldehyde, instead of lysine, it actually prefers the serine. And of course, it actually affected by the composition of the amino acid of the different enzyme. So the glutaraldehyde is actually predominantly interacted with carboxylate ion, and combination of the carboxylate ion with the hydroxy group, and random and uncontrolled interaction were not strong enough. So it is crucial to determine the preferential amino acid by the cross-nuclear. So with that, thank you very much.